All right. Good evening, everyone. Thank you all for being here. Thank you for giving me this opportunity to present my research, which is all about hybrid ligands. Now, if you're like most people, you're probably sitting there wondering to yourself, what on earth is a hybrid ligand? Which is a question I might sarcastically answer, well, clearly it's two or more ligands that have been hybridized together. But this might prompt a further, perhaps more pragmatic question of, well, then what's a ligand? And that is where my analogy comes in. You see, pharmacologists often use the word ligand to refer to molecules that have some pharmacological properties but are otherwise unusable as drugs. Let's say they also have massive fatal toxicity in humans, for example. In a way, you could kind of think of ligands as dead drugs, which would make the scientific literature a dead drug graveyard. So my work involves digging through this graveyard, often late at night, looking for examples of these ligands to hybridize together and create new and interesting molecules. Now, in particular, we're interested in the muscarinic acetylcholine receptors, which are a kind of receptor protein found in your central and peripheral nervous system, which regulate a number of functions such as heart rate, blood pressure, breathing, including brain function. In particular, we're interested in the M1 receptor, because over 30 years ago it was shown that if you can selectively activate this receptor, you can halt the progression of Alzheimer's disease, something I thought was worth working for. Problem is, in 30 years, no one's been able to do this. Now, this is not just because drugs take a long time to go from concept to clinic, but because there's a more fundamental problem with the project. That is, the main binding site of the muscarinic acetylcholine receptors, where all of our most potent drugs bind, is identical in shape in all 5 subtypes. Similarly, we have drugs that bind to alternate sites, and these are selective, but generally not potent enough to be used as drugs. So that's where our idea came in. We thought if we combine 2 of these molecules, an M1 selective and a potent non-selective ligand, we could create a new hybrid ligand which would have the properties of both. The results, however, were surprising and unexpected. Instead of having the predicted M1 selectivity that we'd hoped for, they instead had selectivity for the M4 receptor and now reduced activity for the M1, which, I've got to be honest, was initially quite baffling. So we recalled the hybrids, we dissected up the molecules and analyzed the components again. And what we realized was they were telling us something fundamental about how these drugs work at the molecular level. So the future of the hybrids, although they didn't have the selectivity that we'd hoped for and so won't go on to be the magical cure for Alzheimer's disease, what they told us instead was something fundamental about the molecular mechanisms of selectivity that give rise to the selectivity of ligands like ligand A. An insight which I hope in the future will one day give us the ability to reanimate molecules like ligand A from the dead drug graveyard. Thank you.